Audio Jungle.
Hello, my name is Benis and welcome to our Science Kula episode. And we are going to talk about gene therapy. So what is gene therapy? What is gene therapy? Gene therapy is a medical technique that aims to treat or cure genetic disorders by replacing, removing, or altering the patient's gene. The therapy involves introducing a new gene or genes into the patient's cells to replace, repair, or augment the patient's own defective gene. Did you know there are two main types of gene therapy? Number one is germline gene therapy and somatic cell gene therapy. Germline gene therapy is still in the experimental stage and has not been approved for clinical use. Hey, Chloe, do you know where is Phineas?
I don't know.
Over here! So what is somatic cell gene therapy? Somatic cell gene therapy targets the cells of the body that are not involved in reproduction, such as skin cells, liver cells, or blood cells. The goal of somatic cell gene therapy is to treat the patient's symptoms and improve their quality of life. Somatic cell gene therapy has been more widely used than germline gene therapy as it poses fewer ethical and safety issues. Where the development of gene therapy has been complicated by ethical concerns about the use of human embryos, the potential of unintended genetic changes, and the risk of immune system reactions. Despite these challenges, gene therapy continues to hold promise as a revolutionary medical treatment that could transform the lives of millions of people around the world.
Process of gene therapy. The process of gene therapy involves several steps. First, the therapeutic gene. Therapeutic gene is delivered to the patient's cells using the chosen delivery method. Once the therapeutic gene is inside the cell, it is integrated into the patient's DNA. The integrated gene can then produce the desired protein, which can correct the genetic defect. The correction of the genetic defect can lead to the elevation of the patient's symptoms and potentially cure the disease. Delivery methods. There are several methods used to deliver therapeutic genes to a patient's cells. The most common method is through the use of viral vectors. Viral vectors are modified viruses that are used to carry the therapeutic gene into the patient's cells. The virus is modified in such a way that it cannot cause a disease and will only deliver the therapeutic gene to the targeted cells. Another method is through the use of non-viral vectors. Non-viral vectors are not derived from viruses and are generally safer than viral vectors. Non-viral vectors can be delivered to the patient's cells using various methods such as electroporation or microinjection.